All right, so hi, welcome to the ISME Early Career Community. I'm Gillian Peterson, Chair of the ISME Early Career Scientists Committee, and it's a real pleasure to have actually our president today for an interview that we're starting a series of YouTube video conversations called ISME Conversations with senior researchers in the field of microbial ecology. And we're going to find out a little bit about what it was like for them being early career researchers, how did they get to where they are today, and what could we talk to them about next time we meet them at the ISME party. So we'll get started with Nicole Dubilier. It's a pleasure to interview you today, and perhaps I can still find out something about you after working with you for 11 years. Let's see. Nicole is the director of the Max Planck Institute for Marine Microbiology in Bremen in Germany, and she is a professor of microbial symbiosis at the University of Bremen, head of the symbiosis department at the Institute of Marine Microbiology, and has won all of the awards that are around to win in Germany, in Europe for excellent research. She's a winner of the Leibniz Prize. She got an ERC Advanced Grant and is a member of many academies, European Academy of Microbiology, American Academy of Microbiology, and the Leopoldina Natural Sciences Academy in Germany. So you can see what a pleasure, what a privilege it is to get a bit of her time today. So I'll go straight to my first question. Nicole, so tell me, these achievements are so impressive. How did you get to where you are today? Could you just tell me a little bit about your career path?
Yeah. So first, thanks, Jill, for your kind words in introducing me. Your question, how did I get to where I am now? It was anything but a straight path. It always looks like one was a achiever from the beginning to the end, and my path was certainly not that straight. But in the end, I am where I am today. What I can say is that I was lucky to be at an excellent institute. The Max Planck Institute, or Max Planck Institutes in general, are very well funded, gives you a lot of support financially and also time to really work on your research. And so I really feel very privileged to be working at such an institute.
Okay, so, but you weren't always at a Max Planck Institute. You started out your career, you studied at the University of Hamburg. So what were the steps along the way?
Right, so microbiology was the last thing on my mind. I thought little bugs weren't so interesting, and I wanted to become a marine biologist. And like most marine biologists, I wanted to study whales and dolphins. At some point, I realized that you needed a lot of physics to work with live mammals, and physics was definitely not my strength. And what intrigued me in my studies already at the University of Hamburg in Germany was that in microbiology and biochemistry, which I took next to zoology, was starting to understand how cells function. That part, cellular metabolism, microbial metabolism, I found very intriguing, enjoyed it. Also, the wealth of microbial metabolic pathways was okay, but microbes themselves were still pretty boring. And then I was introduced to chemosynthetic symbioses. My PhD advisor, Olaf Gürer from the University of Hamburg, was working on these little small gutless worms. I remember when, as a student, I found out that he works on worms. I thought, how boring is that to dedicate your whole life to working on worms? And now I work on them. But the cool thing about the worms is they don't have a mouth, they don't have a gut, and they live from their symbiotic bacteria. And that part really attracted me. I found that fascinating. How can that work? How do they feed their host? What does the host give them in return? And together with Olaf Gürer, he is not a molecular biologist, we agreed that I would join Colleen Cavanaugh's lab at Harvard University to look at the molecular biology of the symbiont. So that was my my first baby steps into the field of microbiology. And it was pretty, actually, that was where I discovered my love of microbiology.
Mm. So we're already on the next topic. What is your research about, I guess, and then also the path. How did you decide upon this topic? So how would you describe the goal of your research group?
So the goal of my research group is to understand symbioses between chemosynthetic bacteria and the eukaryotes with which they associate, the marine invertebrates and the marine protists. We have an organismal biology approach. We really try to understand the entire biology of the symbiosis by looking both at the symbionts with which the host is associated, but also the host interacts with its symbionts. We also, let's say, going from the bottom up, we look at the kind of symbionts, the diversity that we find in a host. Then we say, what are they doing? We use lots of omic tools, transcriptomics, proteomics, metabolomics. Then from there, we also move into the environment. We say, what are their energy sources, their carbon sources? How are they interacting with their environment? And then we also tend to look at the ecosystem. So not only a strong focus on the organismal biology, but also the interactions of the organisms with their environment. And then finally, now that we've started to look at the bigger picture long enough, we're starting to also look in more depth at the evolution of these associations, because we're gathering enough data to start to understand how these symbioses evolve.
So it's really a fun, an amazing body of work that your group has produced, and it was a privilege to be a part of that.
It was a privilege to have you.
Thank you.
I very much wanted to have you as a graduate student.
Thank you. So if the goal is to understand the symbioses, the interactions between the host and the symbiont, have you achieved this? Do you think this is
you never achieve your goals as a researcher? No. In fact, one of the things that I truly love about doing research is you answer one question and 300 others pop up. So no, we haven't answered. And well, no, I should say, I believe we've answered quite a few of our questions, but that hundreds to thousands exciting new ones have popped up. And what I also love about working on our symbioses is they are incredibly rewarding in terms of all the cool things we are discovering. We take a descriptive approach in the beginning. We look at their metagenomes, their metatranscriptomes, and metaproteomes, and then say, "Oh my God, look at what's happening." I can give you an example. We discovered about maybe 5 years ago, 10 years ago, that in the gutless worm symbiosis that we work on in the Mediterranean, that one of the symbionts was expressing the genes you need to use carbon monoxide as an energy source. And we said, whoa, carbon monoxide, where's that coming from? And it only took 3 or 4 years to understand that the seagrass peat in which the worms occur, that they are producing carbon monoxide, and that this is the likely source. But these kinds of discoveries that allow us to create hypotheses and then to examine them is one of the most rewarding aspects of our research.
Cool. Okay, so lots of new questions come up, lots of new answers, new topics. I know people have left your lab and have started labs of their own. So what's a question about mentoring? What's the advice you give to your postdocs when they leave and they start their own labs?
Well, there's a German expression that says Ratschläge sind Schläge. And what it means is that advice is actually like getting hit over the head. It's a play on the words. I don't think particularly young researchers leaving my lab want to get advice from me, or maybe the other way around. They're very eager to start their own labs. And of course, just like when I was young, you often think, oh, I think I might even be able to do this better than the lab I come from. What I'm happy about is when I'm then asked for advice. And then that's usually very specific And that's, for example, oh, I have a student and that, how would you deal with that? Or I have a co-author issue, how would you deal with that? And then I feel it's also on a one-to-one discussion at the same level.
Mm-hmm. Yeah, no, I can only say your advice on topics to pursue in my research were extremely valuable.
Thank you.
So we've talked a lot about all the different aspects of the symbiosis that you've worked on. What's your favorite paper? What's your favorite study?
Oh, my favorite. Well, I have two favorites, but I'll start with the one that you're the first author on, which was published in 2011 in Nature, showing that sulfur oxidizing symbionts can use hydrogen as an energy source. And the reason I love it is because it was such a fantastic teamwork effort. We really, and it took us 6 years from the discovery to get that paper published. And we worked on it so hard. And every corner we took, though, we went one step further in really showing that indeed the symbionts can use the hydrogen as an energy source. We had to go on several research cruises. I remember one of the biggest issues was showing that they use it as an energy source for autotrophic CO2 fixation. And then you went on a cruise with Ziege Wechsel, with our technician, and it finally worked and we were able to show that. Then we had the collaboration with Pete Gergitz to measure hydrogen concentrations with his in situ mass spectrometer in situ in the deep sea. So that project, it all just came together, and that was really, that was a true pleasure. And then we got the cover of Nature.
There was some eureka moments in that.
Yes.
That was good times. Anyway, so that's one.
Oh, the second one is my very, very first Nature paper. And that was with the gutless worms I was talking about, the Mediterranean. And I had just started as a postdoc at the Max Planck Institute. And one of the big problems we had when we were looking, what we're trying to find out where the sulfide was coming from, that the sulfur oxidizing symbionts used in these gutless worms. And I was banging my head against the wall trying to find sulfide in the worm's environment and could never find it. And then we were doing very simple 16S ribosomal RNA analyses, then PCR and cloning. Sulfate reducing bacteria kept on popping up in them. And then it took only another 3 or 4 years till we could effectively prove that these symbionts have internalized bacteria sulfate reducers that produce the hydrogen sulfide that the primary symbiont needs. And then together with colleagues at the Max Planck Institute with Nicole Dubilier and Tim Fertelman, we were able to actually show in the worm that the sulfate reducers are producing sulfide by sticking little silver needles inside the worm and showing that sulfide precipitates on them. And then when I, as a young postdoc at the time, went to the directors of our institute and showed them the manuscript, they were only mildly, let me put it this way, they said, well, good luck. And when the reviews came back, and they were very, very positive for reviewers, that was a very exciting moment in my life. And it was the first Nature paper. And then I remember a good friend saying, well, Nicole, one paper is no paper, get used to it. Very soon you're gonna start worrying when you get your second. And she wasn't that wrong. Anyway.
Well, it worked out in the end, right?
Yeah.
So
yeah,
maybe
exactly.
that if, then I'll move on to the question, what was your biggest challenge as an early career scientist? Maybe that was it, or were there other challenges?
Well, first, what is called the rush hour of life. I didn't even learn that expression until a few years ago, but it fits so perfectly when you have a family and then you're also in the middle of trying to build up your career. That was certainly one, probably the 10 years that our son was young. My husband and I both worked full time. That was extremely challenging. And kudos to you, Jill, with 2 children. It's a challenge, and now in the pandemic, particularly challenging. I sympathize with all young parents that are so torn between trying to find enough time for their family and finding enough time for their careers, not to mention having a life in addition.
We will
So
again.
I think in a nutshell, that would be pretty much the greatest challenge. I would say after that, the challenge was figuring out for myself if I wanted to move from postdocing forever or a more, let's say, a position where I had a lot of research independence but did not lead a large group, into did I want to take this next step to become first a group leader and then a director.
That's interesting. So was there a moment where, or an event that pushed you in either direction?
Yes.
Or? Mm-hmm.
When a colleague of mine that I thought was not as qualified advanced, a male colleague, no less, I do think that I was naive in retrospect. Not naive, maybe that's actually too dismissive. I think that the advantages that I had as a postdoc in the Max Planck, where I was independent with my research and where I was also, many thanks to Rudy Ammann, whose lab I was in at the time as a postdoc, to allowing me to have a very small but very enthusiastic group. There's a lot to be said for that. I also had no responsibilities, no administrative responsibilities. So it was really a golden era. But then to see others advance, have larger groups, be able to take on more research projects. At that point, I said, wow, you're always going to be here. And you're going to get older and older, and you're going to watch others advance past you. And at that point, I said, well, why don't you just try it and see if you like it? And that was actually an excellent step forward, was just to say, you can always go back and become a postdoc again. I mean, I think that's actually rather hard, but at least giving myself that room.
Mm-hmm. Going easy on yourself.
Oh yeah, always a good idea.
I'm sure I've got a question about that. Yeah. One piece that you do give advice. It's more like in the context of just talking about life. There's one thing you always told me, is that look after the golden goose.
Oh yeah.
You don't take care of yourself. What hope do you have of managing a research group and managing research projects? So what do you do to relax?
Well, I used to take very long vacations. Certainly when our son was younger, just taking time off was incredibly important. Now it changes from year to year. For a while, I tried mindful meditation. That was very important. There was a time when I felt that I wasn't able to relax well, and doing mindful meditation was an excellent way for me to discover the type of relaxation I need to then come back energized to work. Lately, since the pandemic, it's been jogging. That just refreshes my mind. And then critical for me is spending the evenings with my husband. Now there's no company, but with friends and family, and enjoying time with them, sharing good meals with each other. Nice. And good wine, of course.
Yes. Can't wait to share a wine again with you.
Oh, yes. In person, not virtually.
Absolutely. Maybe coming to the last question, talking about relaxing and unwinding. What's your favorite ISME memory? What's your favorite ISME memory? Right?
Oh, that is Fantastic question. It's always the last party. ISME parties at our in-person meetings are really wonderful. Everyone that's attentive one knows how much fun they are. And so I could, of course, be more diplomatic and say the fantastic science I hear at every meeting, and that is in fact essential to the meeting, but the parties are fantastic.
I agree. Maybe this is a good point to explain the noise coming from my background is my 10-year-old daughter in homeschooling. So thanks everybody for putting up with that. Speak of the devil. So anything else you'd like to tell the ISME early career community in parting?
Maybe I just said I don't give advice, but now maybe to early career.
No, you've been asked specifically.
Oh, okay, thank you. I think that one of the most important qualities to have is persistence. I don't think that all the other qualities, you have to be highly intelligent, you have to be highly creative, you have to be highly innovative. That I found discouraging because I always was able to find people that are smarter than me, always able to find people that are more creative than I am. The list is endless. But I do believe that my persistence was extremely helpful in allowing me to continue down my path. That I always said, you know what? For example, my ERC grant was rejected twice. The third time it was accepted. This, I think, is a very important quality, and I recommend that every early career researcher, no matter how often things don't work out, just be persistent. In the end, they will.
Great advice. Thank you very much. So thanks for your time, Nicole. This has been really fun. And we look forward to the next installment. We're releasing these once a month approximately, so look out.
Yeah, wonderful idea. Thanks for setting that up.
It's fun for us too. Okay, bye.
Okay, bye.